Hi, everyone. Welcome to the Behavior, Evolution, and Culture speaker series here at UCLA. This is the last talk of the 2020-2021 academic year. And then we'll be taking a break for the summer and returning in fall. So I don't have any announcements of upcoming talks because we still don't have the schedule set for fall, but as that becomes available, we'll be posting the schedule on the BEC website, which is bec.ucla.edu. There, of course, you can find a recording of this talk as well as an archive of past talks. You can access that either through the BEC webpage or just directly through the YouTube channel, which is UCLA BEC, all one word. Today, it is my great pleasure to welcome Professor Alyssa Crittenden from the Department of Anthropology at UNLV. And we are welcoming Professor Crittenden back because she has given a BEC talk before. What year was it? It was probably maybe 10 years ago now or something that you did your last talk.
Yeah, that sounds right. 10 years, maybe, give or take.
Yeah. So it's great to have you back, Alyssa. Welcome. And Alyssa's talk will be called Microbiomania, Rewilding, and the Threat of Bioprospecting: How Anthropologists Can Help Set a More Ethical Research Agenda in Microbiome Sciences. If anyone would like to unmute themselves to join me in welcoming Alyssa, please do so. Welcome. Welcome, Alyssa. Go ahead and share your screen and get started whenever you like.
Great, thank you so much. Okay, let's see if that works. How are we doing? Clark, thumbs up. Great, all right, thanks.
Yeah, I can see it. We can see the main screen, yep.
Wonderful, thank you so much. Good afternoon. Before I begin, I'd just like to say thank you. Thank you to BEC, thank you to UCLA, and thank you to Clark for the invitation. I'm really looking forward to speaking to everyone today. Before I begin my talk, I'd like to provide a short biography. I'm speaking to you from Boulder City, Nevada, just outside of Las Vegas, and from the traditional lands of the Paiute and the Mojave peoples. My orientation is as a non-Indigenous female scientist with training in nutrition, ecology, and biological anthropology. Given the increasing spotlight on biological anthropology as a discipline which was founded in settler colonialism, I think that this explicit acknowledgement at the beginning is warranted. My research over the past 20 years has focused on the intersection of nutrition, behavior, and childrearing in small-scale Indigenous communities. I've worked with the Hadza foragers in Tanzania since 2004, and I was last in the field just a few months before the COVID-19 pandemic hit. I've collaborated on work exploring diet composition and the gut microbiome, which will be the overarching theme of my talk today. I no longer engage in microbiome work that demands sample extraction, something that I've discussed in detail elsewhere and something that I'm happy to talk about during the question period. I'm also presenting some collaborative work that's in revision at the end of the talk, and some work on rewilding specifically that's in preparation and has been commissioned for a forthcoming perspective piece. The bulk of the talk, most of the talk, is based on the two papers that you see on the screen here today, in case you would like to read further about some of the orientation and the motivation for this talk today. I welcome feedback, and I'm hopeful that sharing some of these ideas with you today will make for stronger argumentation in the future. So what am I talking about today? Today I'm going to be broadly discussing how microbiologists are turning to ecology, anthropology, and human biology to inform their discourse on rewilding. And this provides an opportunity for provocation. Today I'll attempt to answer the following questions. What are microbiome scientists getting right? What are they getting wrong? And how does this impact study communities? And I'll end with a discussion of how anthropologists can enter into the discussion. So I offer a brief outline of my talk here. I'll introduce the concept of microbiomania and the microbial turn in human biology. I'll discuss the concept of old friends shifting to missing microbes. We'll talk about rewilding in particular, the gut microbiome of small-scale societies and what the science has told us. We'll discuss the quote-unquote ancestral microbiome, how we rewild and who benefits from this process. I'll introduce a framework, political ecology of the body, and then lead that into a discussion of biopower and the threat of biopiracy. And finally, I'll end with hopefully helping to answer the question of where do we go from here. The human microbiome is the full collection of genes of microbes and microorganisms. A microbe is a microscopic living organism residing in and on our bodies, and I'm sure most of us are aware of this, but just as a very kind of brief overcap, each one of us hosts about 40 trillion microbes on and in our bodies, and they act to defend us from pathogens, boost and protect our immune system, keep infants healthy, and the list goes on and on. It's hard to turn around today and not run into another advertisement, news article, or study involving the gut microbiome and its role in human health. As our understanding of the vital role of microbes increases exponentially, so too does microbiomania, the fervor in which microbes are lauded in popular media and scientific press as capable of revolutionizing human health in the global north. The microbial turn in biology is when bacterial species went from being considered threatening to beneficial. Based on Louis Pasteur's work, we learned that vaccination could be applied to any microbial disease. Through his work, we learned that the virulence of microbes could be attenuated so that live microbes could be used to make prophylactic vaccines. He was working at the dawn of microbiology, and the war on microbes included washing, sterile surgical procedures, and of course, antibiotics that saved countless lives. Microbes were seen as the enemy, and they were to be eradicated. The profound success of these germ-killing techniques and drugs over the last century, with minimal acute side effects, has led to their overuse. We now know, however, that limited exposure to microbes may result in defects in immune function or regulation. So this can lead to increased burden of autoimmune diseases. And early iterations of the hygiene hypothesis, which was originally introduced in the late 1980s, argued that early childhood exposure to particular microorganisms, like gut flora and helminth parasites in particular, could act to protect against allergic diseases by contributing to the development of the immune system. The hygiene hypothesis has now been extended to account for the role of various parasites like intestinal helminths and microbiota compositional shifts. Both of these extensions, so the old friends hypothesis and the microbiome depletion or the microflora hypothesis, provide explanation for how environmental exposures act to shape the human immune system, and how microbes have gone essentially in the dome of biology from peril to promise. And this is an important setup for the whole concept of rewilding. Okay, so how did we go from old friends to missing microbes? The basic premise of the old friends hypothesis is that contact with old friends, so relatively harmless microorganisms that have been present throughout mammalian evolution, is greatly diminished in urban, sterile environments. So the context in which most of us live, the context in which I'm speaking to now. If you live on a farm, if you keep a lot of pets, if you play in the dirt, you may increase exposure to these old friends. Helminths are one of the most heavily studied of the organisms that likely co-evolved with humans and other mammals. And it's been argued that they've likely exerted considerable selection pressure on our immune systems. In this case, helminths, as an example, are friends because our immune systems tolerate them because attempts at elimination can lead to immunopathology. So these old friends, of which helminths are one example, are implicated in many evolutionary mismatch arguments. This has been recently extended to missing microbes, where the absence of certain keystone species in our gut microbiome has ostensibly caused the system to go awry and has led to dysbiosis, or an imbalance of microorganisms on or within our bodies. Microbiologists using a variant of the old friends hypothesis now argue that these missing microbes might account for the dramatic recent increases in allergic, inflammatory, and autoimmune diseases seen in populations in the global north. So our microbiome arguably needs to be repaired, as the story goes, and these missing microbes reintroduced. And this is the premise for the rewilding movement, the so-called rewilding of our guts. Before we focus full tilt on rewilding the gut microbiome, I want to introduce the concept more generally. Rewilding as a term was originally tethered to environmental science and conservation biology, and it's been co-opted numerous times across multiple literatures, including conservation activism most recently. Many definitions abound, but a general one introduced by the Society for Ecological Restoration several years ago is, quote, the process of assisting the recovery of an ecosystem that has been degraded, damaged, or destroyed. So nearly all scientists who discuss rewilding as a concept agree that they're trying to reinstate something. They just can't seem to agree on what that something is that they're trying to reinstate, other than wildness. There are no agreed upon definitions, which is partially responsible for how the concept has been wholeheartedly co-opted for human health in recent years. The popular conception of rewilding, as you see embodied here by a Facebook group in the lower right-hand corner of the screen, is one very popular approach. The same applies to the language used in a popular podcast called Rewild Yourself, which is led by the host of a TV show called Wild Fed. The concept is everywhere, it's pervasive, and it's captivating. But it begs the question, as human geographer Dolly Jorgensen states, what does it mean to be wilder? Wilder than what? And this is what we'll be talking about for the remainder of the talk today. While increasingly the term rewild is used in microbiology and in the popular media in regard to the microbiome, it's rarely defined. It's largely applied in two contexts, natural landscapes and human bodies. In terms of natural landscapes, it's largely tethered to restoration ecology, which we don't have time to get into today. But data have shown that when you increase plant species diversity, you also increase the diversity of the soil, which increases ecosystem function, or can potentially increase ecosystem function. Evidence suggests that it's also possible to increase ecosystem function by inoculating soil with particular strains of bacteria that enhance the establishment of key plant species while simultaneously increasing soil fertility and also quality. In addition to natural landscapes, there are also a lot of pushes right now to rewild the human body. So the second place in which rewilding is occurring, or attempts at rewilding are occurring, are the human body, whether it be through diet or lifestyle. Rewilding the gut microbiome involves the goal of restoring the gut microbiome, or the GM, to its quote unquote ancestral or healthier state. And we'll be unpacking these terms for the rest of our talk today, my talk today. So how do we do this? There are a few pathways that are lauded in the literature. One is to change your diet, to take supplementation or engage in other therapies like fecal transplant, or reintroduce the missing microbes to our dysbiotic guts. And this involves replacing lost gut microbial taxa or engineering microbes to perform depleted functions, or transplanting whole gut microbial communities from donors to recipients. All of these are pathways to rewilding. So revisiting Jorgensen's provocation of what does it mean to be wilder than what, the real question as it pertains to rewilding the gut microbiome is whose gut is the most wild? In 2017, a researcher from King's College accompanied an autodidact and self-proclaimed anthropologist to Tanzania and spent 3 days eating a hunter-gatherer diet, eating only wild foods collected by Hadza foragers. He then returned to London and had his feces analyzed and argued that his gut health was indeed improved, at least in the short term. But what did he mean when he said his gut health was improved? And why are these guts the targeted wild guts? We'll be unpacking this in a few moments, but first we have to establish who we are rewilding ourselves to and why. Contemporary hunting and gathering populations are often used in many evolutionary models, as I'm sure all of you are very well aware, given the crowd that I'm talking to today. How one defines or classifies a forager, however, may be based on food economy, social structure, or any other number of given traits. As Richard Lee aptly stated many years ago, Even if it is agreed upon that hunter-gatherers exist, almost everything else about them is a matter for contestation. And this is certainly true today. There are some characteristics, however, that are considered to be key traits of foraging communities that are central to many models of nutrition and health. And these traits include diet that includes non-domesticated foods to varying degrees, depending on where you are on that spectrum of a mixed subsistence diet, limited or no tending of crops or herd animals, mobility, so residency patterns that may shift to track resources, again, also existing on a continuum. Relatively low population density, although this certainly depends on the population, and minimal access to biomedicine, also depending on the population. But regardless of what distinction or what definition is used, contemporary foragers continue to be used as referential standards for inferring evolutionary origins of behavior, health, and diet. While hunter-gatherer studies have waxed and waned over the last several decades, their interest to evolutionary, sociological, and demographic and human health science studies is having a resurgence right now. And this is clearly the case with the hunter-gatherer gut microbiome, which is often seen as a proxy for the ancestral microbiome. Collaborative work that I've been part of, as it turns out, is a central part of the story. In 2014, my colleagues and I published the first report of gut microbiome data from a foraging community. My colleague and longtime collaborator, Stephanie Schnorr, collected fecal samples as part of her dissertation work at Max Planck Institute of Evolutionary Anthropology when she was a doctoral student and we began our collaboration. And I should use this opportunity to talk about the work that Steph did to remind all of us that from here on out, given that this is a talk about the gut microbiome, we're going to be talking a lot about fecal samples. So we're human biologists, we're evolutionary anthropologists, so hopefully this is not a problem, but I'll try to give you a heads up if it gets, there's a couple of pretty exciting graphic quotes that I'll read, and I'll give everybody kind of an alert in case you want to tune out for a few minutes. But for the most part, we'll just be talking about fecal sample collection for the next few slides. So when Steph initially went out and collected the samples that provided the basis for this first study, our initial goal was to map the species that were there. So essentially just to map the colony that Hadza guts in general were housing. We ended up publishing several other studies, but this was the first one, and it was one that demonstrated some really interesting trends. First of all, as you can see here on the screen, when comparing the Hadza data in the upper left-hand quadrant to other populations for which we had comparative data at the time, this will be an important caveat moving forward, the Hadza configuration of gut bacteria was more diverse than any population that had been previously sampled and published. This is no longer the case. And it turns out that the Hadza indeed fall along a gradient, not only of foragers, but of other subsistence level societies. We also learned that Hadza house certain species that are known carbohydrate metabolizers. So this includes non-pathogenic clades of treponema, which makes sense based on Hadza diet, which contains a large amount of tubers or underground storage organs. In the intervening years, there have been so many additional studies, over 60 of them since we initially published this paper, that we can now identify trends in the hunter-gatherer gut microbiome or in the gut microbiome of small-scale non-industrial populations. Populations sampled have included foragers, pastoralists, and agriculturalists. All are subsistence level societies and consume wild or locally harvested and minimally produced foods. The big overarching takeaway is that lifestyle and dietary factors exert a profound effect on gut microbiome configuration. Here in a paper published recently in Trends in Microbiology in 2019, you can see how subsistence strategy is classified with general traits and associated taxa and functional features listed. In foraging communities that consume high fiber diets, you see enrichment in Prevotella and Treponema species, which work to metabolize complex carbohydrates. The amount and type of carbohydrates in the diet differentially affects the abundance ratio of short chain fatty acids or SCFA production, particularly propionate SCFA, which is a byproduct of bacterial fermentation in the large intestines and has been associated with cholesterol lowering, reduced fat storage, and in some instances, anti-inflammation. At the other end of the comparative spectrum, we've found that urban dwelling individuals house colonies of gut microbes that contain little or no Prevotella and are characterized by very little diversity. Lifestyle is also characterized by antibiotic use, which is known to deplete colony diversity even after one dose. In the intermediate space between foragers and urban industrialites, small-scale agriculturalists are slotted in, making a very easily understood, ostensibly easily understood, clear evolutionary argument. And I will argue it's the appearance of a very clear evolutionary argument, but this is the typical story. This is how it's typically laid out, and we're going to unpack it. Of course, it couldn't be that easy. If it were that easy, I wouldn't be here talking to you today about this. So we know that there's more to the story. So what do we know after such an intense deep dive into microbiome research over the last decade? We know that microbiotic composition, diversity, and gene content in the microbiomes of small-scale rural communities vary substantially from populations in urban centers. This means that very broadly, more microbial exposure correlates with a more diverse gut microbiome, using really big brush strokes. And this has been well established. Each new study confirms the lack of diversity in the GM of most sampled populations in the urban dwelling global north. We also know that many subsistence level societies, regardless of ecology or food economy, share some taxa that are notably absent in North Atlantic populations. And I should say North Atlantic urban guts. Urban's an important component here. These taxa have been dubbed vanish taxa. The acronym stands for volatile and or associated negatively with industrialized societies of humans. And this term was introduced by Justin and Erica Sonnenberg and colleagues at Stanford. More on that momentarily. But here's where it gets interesting. So we don't see vanished taxa in all mobile or semi-mobile subsistence populations who are consuming wild or foods that aren't processed very heavily, which we would expect to, based on the map I just showed you, the graphical abstract, that this is kind of a very clear linear argument and that you have a microbiome that associates with subsistence. One example is a recent paper profiling the taxonomic, essentially the colony, the intestinal microbiome of ethnic Mongolians. And their gut microbiome did not contain any vanished taxa, as we would have expected based on the simplistic model that I showed you in the last slide. Rather, their diet, which contains significant amounts of red meat and locally minimally processed cheese, so unpasteurized cheese product, their diet is reflected in their gut microbiome. So this means that it's high in genera that are associated with metabolism of animal protein in particular, and a variety of amino acids and saturated fats. So while some of the microbiome similarity that we see across these subsistence level populations that go into creating those graphical abstracts that tell a very clear-cut, ostensibly clear-cut story, we know that this has to do with similar nutrition profiles. And this may largely be tethered to things like the inclusion of dietary fiber. So populations with high meat eating exhibit colonies that don't contain vanished taxa, and those who eat high fiber diets are going to essentially, with similar nutritional profiles, are going to have the same microbial exposure and may indeed share some of these same taxa. It's also noteworthy, it's also, I think, very important to point out, which doesn't often get included in any of the models, that some key genera like Prevotella, which is considered one of these keystone species that we hear about a lot, are not found only among foragers. They're also found in the guts of individuals in urban centers around the world who eat plant-based diets long-term. And in a recent study where researchers from the global north consumed a wild food diet for 16 days, their gut microbiome more closely resembled the GM of the study population living in rural Venezuela. Okay, so what does all this tell us? The takeaway here is that gut microbiome data from small-scale populations tells us that community assembly is different. It varies based on diet composition and exposure to environmental microbes. something confirmed by data from hunter-gatherers, but certainly not driven by it. All right, so what is the ancestral microbiome? If hunter-gatherers share gut microbiome traits with other non-foraging groups who have similar levels of microbial exposure and dietary similarities, then what exactly is the ancestral microbiome? The ancestral microbiome is the non-modern microbiome, as it's articulated both in scientific press and lauded in popular media. This 2019 article published in Frontiers in Microbiology refers to contemporary hunter-gatherers residing in Africa and South America as ancient. And this is essentially part of, this is the crux of it right here. So traditional is associated with non-modern, which is associated with ancient, which then becomes associated with ancestral. The notion that traditional actually means ancestral is a contemporary spin on primitive. And it's outlined beautifully in a forthcoming paper by your very own Clark Barrett. In his ancestral gambit argument, he states that we're often looking to hunting and gathering communities as people who we think are living under circumstances, ecological, social, or cultural, that most resemble ancestral lifeways. And while this certainly might be true, depending on the variable of interest, it is often not true. And as Clark fleshes out in his full treatment of the subject, I then extend this to the gut microbiome. Contemporary foragers, particularly for disciplines outside of anthropology these days, are viewed as ancestral. As the previous slide indicated, contemporary living people were described as ancient. This is partially based on mode of subsistence, but it's really a high fiber diet and more microbial exposure that largely leads to a diversified colony of gut microbes. And the so-called missing microbes, the vanished microbes, those are present in other high microbial exposure non-foraging refugees who've been displaced from urban centers, and they're also present in small scale, non-industrial populations like Mongolians, where you would anticipate to see these vanished taxa present, and they're not there because they are adapted to their diet and to their ecology. So why is the hunter-gatherer microbiome the best microbiome? And the answer is because it's ancestral. And ancestral is also thought to be closer in line with our evolutionary past and therefore healthier. We'll address the myth of the healthy microbiome in a few minutes, but first I'd like to talk about why this language is dangerous and laden with colonial nostalgia. Hunting and gathering communities, due to their diverse microbial gut colonies, are billed as presumably closer to a healthier and disappearing ideal. There are many issues with this line of thinking. First and most significantly, there are no data to suggest that a forager gut microbiome is indeed healthier. Many pushing for the argument that we're missing keystone species in our gut, the species that we need to save from extinction, are basing the notion on the idea that communities, like the Hadza, are representative or are a healthier ideal that is vanishing, just as the microbes that they gave the same moniker to. This statement in a recent 2019 piece in Science states that components of the microbiome that lived within humans throughout most of our existence as a species likely promote biology consistent with a long and healthy life. It's the second quote on the screen. The citation for this claim is Frank Marlowe's 2000 article, The Patriarch Hypothesis, which does indeed discuss longevity, but has absolutely nothing to do with the gut microbiome. Despite claims that our microbial identity has deteriorated and is suboptimal for our health, what you see stated in the top quote, we have virtually no data suggesting that an ancestral microbiome, if we could identify one, is healthy. Just in the last 10 days, so hot off the press, and you see this is the Nature paper seen in the screen on the right-hand side, analysis of fossilized feces from 2,000 years ago in the southwestern US and Mexico was published in Nature. These data indicate that mobile subsistence level communities sampled had gut profiles that were similar to some subsistence level societies who consume largely plant-based diets today. Well, this may be seen as by some as confirmation that many hunter-gatherers do indeed have something more akin to an ancient microbiome. We must interpret these data with extreme caution. Using computational tools to predict information about proteins encoded by DNA for previously unknown organisms, which they identify in this study, is challenging to say the least, particularly when making claims about a healthy gut. And it's also critical to mention that microbiomes are highly variable between individuals and between populations. And this is what we'll turn to next. Leaving the lack of data aside, we can focus on what we do know. Humans, like other animals, are holobionts, an assemblage of the host and other species living in and on it. The host, its virome, its microbiome, all contribute in some way to the function of the whole, and together form a discrete ecological unit. As Donna Haraway put it years ago in a different context, this means that human nature is an interspecies relationship. So too, I argue, is human biology. The hologenome, then, is the collective of genomes of the holobiont. In one recent model, pictured here on the right, holobiont phenotypes can change in time and space as microbes come into and out of the holobiont. Microbes in the environment are part of the environmental metagenome. Hologenomes then encompass the genomes of the host and all of its microbes at any given time point and are also acquired from the environment. The debate as to how selection acts on the hologenome is a fiery one, and it's currently underway. The take-home message here is that even if it were reasonable to assume that the hunter-gatherer microbiome is healthier, which we don't know that it is, because it is similar to that of our ancestors, which it likely isn't, we could not rewild to the microbe of hunter-gatherers even if we wanted to. Our microbiome encompasses our life history. Microbes may be transmitted vertically in utero or horizontally between different members of an ecosystem. They're acquired from the environment and are adapted to and impacted by our diet, our history of medication use, pathogen and viral burden, the water we've consumed, whether or not we have pets, and the list goes on and on. We lack any theoretical or empirical consensus on these points, making the concept of rewilding faulty from the outset. We cannot say what this wild referential state is, or whether or not it would indeed be healthy for anyone given ecological circumstances. But let's just say, for argument's sake, that you could rewild, that health would improve upon restoring our guts to some pre-industrial or ancestral microbiome state. How would we do this? How does one do this? Microbiologists have zeroed in on three basic categories of rewilding mechanisms. One is to replace these missing microbes. Another is to engineer microbes to perform beneficial functions. And the third is to transplant communities from donors in traditional societies. The image that you see on the screen is from a 2017 article, and it specifically references data from small-scale non-industrial, very living, very contemporary human populations that are then categorized with the image on the far left where it says past. I draw your attention particularly to the club that's being held by our forebear there. Okay, so how do you rewild your microbiome? The first is to eat a wild diet. So you can see here, I really like this one from the Hindustan Times. It hits close to home. Should you follow the Hadza diet? Even if you could, should you? The other, which we'll talk about in more detail momentarily, is from a paper in the UK, The Conversation, written by Tim Spector. I spent 3 days as a hunter-gatherer to see if it would improve my gut health. So the study that we've already talked about. So you can change your diet. And many of you can actually access baobab powder, should you want to, at your local health food market. So you too can eat like a Hadza person. We're going to get into a certain nutrition bar that you may be able to get your hands on soon. The other option to rewild your gut microbiome is to repopulate your gut. And I pull out this interview in Nautilus, although these quotes are ubiquitous. I pulled these out particularly because they offer some really good fodder for us to unpack as we move to the last portion of the talk. It's really our last chance to harvest a lot of these microbes from around the world. We have to do it before it's too late, and it's very nearly too late. And these are from some of the lead microbiologists in the discipline. And I don't mean this as an indictment, because I think that part of the responsibility, as I'll argue at the end of the talk, falls upon us as anthropologists to intervene in dialogues of this nature and call it out when we see it. But here we are. So such communities house a kind of microbial ark, a storage vault for potentially endangered human microbes. So this microbial ark is what is argued is needed for global health enrichment. We may have to repopulate ourselves with microbes derived from more traditional living populations, from, say, Amazonian Amerindians or African hunter-gatherers. Okay, so we're going to move into how one would repopulate their gut, not using diet. And this is the part of the talk where we'll be talking about fecal transplants. So if you are squeamish, now is your time to duck out. Arguably, we're entering the spiciest part of my talk for today. It's the spicy hot wing section of my talk. And given that we're about to talk about fecal transplants, I think that this is a very apt moniker. I'm going to give you a very brief overview of this episode, and then, time allowing, let me check the clock, read aloud from the book that you see pictured on the right, Rewild, which chronicles the story that I'm about to share. You can see some of the headlines. This was a huge splash in 2014. For those of you who don't follow either Hadza literature or gut microbiome literature or the media, you might have missed it, but it was a huge story. Whether or not this particular event actually happened is still up for debate. But at the time, it was covered everywhere, including in Science magazine, as an event that happened. The New York Times covered it, Science covered it, every popular media outlet covered it. I also would like to direct your attention, before I read the excerpt from the book, to the image that you see in the middle, the human food bar. This is an advert. I have the book right here with me. So here's the book. And here's the advertisement for the human food bar that you see right there on the screen. And this will allow you to do the first mechanism of rewilding your gut microbiome. This bar is no longer sold. It was for reasons that I'm unaware of, but you're 99% microbe, which is actually also, we've overturned that since then, but it's time you start eating like it. No artificial preservatives or freaky stuff of any kind. You heard us. It contains, I'm assuming I think that's an ingredient list on the bottom, prebiotic fiber. It's gluten-free. These are characteristics of the bar. It's grain-free. It's Hadza food. And my favorite ingredient is science. So this bar contains science. All right, here we go. Okay, so what you have here this was kind of what kicked off the rewilding movement. The book is called Rewild. It's incredibly popular. And it's kind of like, this is the story that launched 1,000 podcasts. And it was a scientist, a researcher working with the Hadza named Jeff Leach, who gave himself a fecal transplant to try a Hadza gut microbiome. And I'll just read the very first chapter, because I decided to read it as opposed to putting it on the screen, because too much text and we might lose some of the context here. As the sun set over Lake Eyasi in Tanzania, nearly 30 minutes had passed since I had inserted a turkey baster into my bum and injected the feces of a Hadza man, a member of one of the last remaining hunter-gatherer tribes in the world, into the nether regions of my distal colon. I struggled to keep my legs in the air with my toes pointing towards what I thought was the faint outline of the Southern Cross rising in the evening sky. With my hands under my hips and butt perched against a large rock for support, I pedaled an imaginary upside-down bicycle in the air to pass the time as I struggled to make sure my new gut ecosystem stayed put inside me. With my butt cheeks flexed and my you-know-what puckered, I wondered if I had just made a terrible, terrible mistake. Could I really displace my Western gut microbial ecosystem with that of a man who only days before had dined on animals as diverse as zebra and monkey, and who possessed one of the most diverse gut microbiomes of any person on the planet? Would my immune system soon freak out at the presence of what should be some familiar old microbial friends now setting up shop throughout my slimy vastness of my gastrointestinal tract? or had I just unwittingly infected myself with some lethal bacteria or virus? The pros and cons, mostly cons, of my turkey basting activities raced through my anxious mind as I pedaled my way into the evening. I read this section in particular because I have been asked countless times to talk about this in academic contexts, in conferences, when I'm doing public media outreach, in museums, in elementary and middle schools, where I do some of my NSF EBSCOR work here at UNLV. This story is pervasive and it kicked off a movement. Whether or not this scenario actually happened, I don't know. At the time, it was treated as an actual fecal transplant. Okay, keeping that story in our mind. I argue that contemporary foragers in the microbiome movement, as just articulated from the book I read a snapshot from, I argue that contemporary foragers right now are 21st century reiterations of the noble savage stock character. So the noble savage, long known to us, both from fictional and scientific works over the last century and beyond, is an individual untouched by civilization and therefore uncorrupted, often argued to be closer to nature and therefore closer to the inherent goodness of humanity. With the move to decolonize the academy, most certainly reverberating through anthropology right at this moment, as we are all aware, there is a move away from overt characterization like this. That doesn't mean, however, that the noble savage is not being employed as a trope. Here I provide two examples of how foragers, in this case the Hadza, are used in product placement. It's quaint to see foragers in the bush reading labels and book chapters. So on the bottom, you see an individual reading the label for the human food bar, and on the top, you see an individual reading a book about the gut microbiome. Never mind that they don't speak English and are largely non-literate. Beyond the colonial and racist undertones of images and actions like those depicted here, no matter how unintentional, there is a broader context often undiscussed about axes of power and relationships between those studying and those being studied. Beyond the critiques of the scientific validity of rewilding, I'd also like to discuss the movement through a framework that it's based on the political ecology of the body, or PEB. The PEB framework allows for a more nuanced discussion of how labeling some communities as ancestral and non-modern can have policy implications. PEB interrogates global health discourse and emphasizes that health is shaped through the relationships between social and environmental systems. Importantly, it centers on power dynamics, the power dynamics of who decides what a healthy body is, whose bodies are healthy. Many political ecologists are associating recent health trends with Foucault's concept of biopower, most basically defined as having power over bodies. And I use this framework to explore the unintended consequences of labeling hunter-gatherer microbes as ancestral and simultaneously vital. What does it mean for the donor populations when the individuals extracting the biological samples are the ones in power, from powerful countries and driving policy decisions? With calls that we may have to repopulate ourselves with microbes that are derived from, say, in this quote, Amazonian Amerindians or African hunter-gatherers, the question becomes who controls the use and distribution of the species that are in this proverbial microbial arc, or that will be placed into it. Who benefits? The answer presumably is that all of us benefit. Us being the people on this talk today. Those of us with industrialized gut microbiomes that are mismatched with our evolutionary past. Those of us with dysbiotic guts can one day, in the hypothetical not too distant future, pop a probiotic that is derived from species extracted from the feces of an indigenous person. These species are canonized in the equivalent of a seed bank or an ark that can be tapped when needed. So who benefits from this microbial arc? Access to basic antibiotics is already limited in many of the communities who house these species. They certainly don't need a prebiotic to introduce those particular species back into their guts. They certainly haven't lost them yet, so they're not benefiting. With escalating climate change and increased market integration and increased dietary change that is characterizing populations who are transitioning away from traditional food economies, such communities may benefit from other probiotic interventions or from antibiotic intervention. But that's not what's driving this movement. It's driven, as Jamie Lorimer, a human geographer, has aptly stated, by political and economic elites of the global north. So it's a clear example of biopower, whether or not we choose to articulate it as such. Axes of power are central to this discussion of the microbial arc. There's a clear threat of biopiracy in this movement, often disregarded as the worst case scenario. I had a reviewer recently say, point us to an example of where this has happened. We found one. It's happened on countless occasions over and over and over again, often with drugs and cosmetics, but also with bacterial strains. In the essence of time, I'm only going to provide one example to highlight the very real threat of biopiracy in this movement. Biogaia is a Swedish biotech company that holds a patent on a bacterial strain named Lactobacillus reuteri protectus. Biogaia researchers were looking for a bacterium in the early 2000s to use as a new probiotic in infant formula. And they settled on Lactobacillus reuteri, which is a known bacterium with antibiotic properties. Their head scientists went to Peru to a rural village in the Andes and collected breast milk samples. These were then analyzed and deposited in a biorepository and tagged as ATCC PTA-6475. I include that here in the event that anyone wants to look up the history of the patent, which is publicly available. This strain, dubbed Lactobacillus reuteri protectus, was classified on the patent as a, quote, biologically pure culture of Lactobacillus reuteri. This pure culture was then sold to Nestle for infant formula, put into perpetual license for 50.8 million euros, over $60 million. Well, the participants were consented and they agreed to sampling and were compensated. There is no information available on whether or not they have been additionally compensated, and Nestle and Biogaia have declined to comment at every opportunity over the last decade. It should be noted, however, that they did nothing illegal. It is not required to engage in a profit sharing or benefit sharing agreement from populations who are sampled. So where do we stand with rewilding after all of this? This leads us to the point where we need to take stock of everything I've just talked about. So what does the science say, and where do we go, given the challenges and power dynamics that I just outlined? The first point to make is that although there still may be very good reason to include hunting and gathering and other subsistence level communities in cross-cultural samples, we need to be more nuanced as to why we're including such communities in the first place. This speaks directly to the ancestral gambit concept introduced by Clark Barrett. Context matters. Without it, arguments that contemporary peoples house ancient ecological systems go unchecked. In terms of the science, we know that varying degrees of microbial exposure and diet composition alter microbial communities in the gut. And this is confirmed with data from foraging societies, but again, by no means driven by it. There is also no one ancestral microbiome beyond the fact that our ancestors did indeed house a gut microbiome. We have no data to suggest what this might look like. There is also no agreement on what a healthy microbiome is, given that humans are a holobiont. Our microbiomes are impacted vertically and horizontally, and they're impacted by environmental exposures, lifetime patterns of diet and medication use. Even the people and animals who we come into contact with impact our microbiome. As Seth Bordenstein recently stated, biologists are just starting to get a handle on basic questions of microbe-host interactions. We have a century's worth of work ahead of us to figure this out. This makes discussion of rewilding not only scientifically inaccurate, it makes it premature. We must also interrogate the ways in which study communities are being presented and why this matters in terms of policy decisions. Power dynamics are always at play, and the individuals who prosper are not the ones who have provided the biological samples in question. This speaks not only to colonial nostalgia and a reimagining of the noble savage paradigm, but it's unethical. And given the move to decolonize the academy, calling out power differentials matters. So where do we go from here? As our understanding of the vital role of microbes increases, so too does the potential for microbiome research to offer health intervention, largely to those of us who are affluent and reside in the North Atlantic. So this necessitates a transdisciplinary dialogue of the ethical challenges facing researchers who work in the microbiome sciences. And this necessarily includes anthropologists like myself. So where do we go? Given that anthropologists and their scholarship are increasingly implicated in cross-cultural studies of the microbiome and in the rewilding movement specifically, we need to enter into the conversation. I am no bioethics expert, I only dabble, but I can offer my perspective based on lessons learned and taking cues from the existing scholarship on what a more ethical research praxis with Indigenous communities might look like. The first step, at least in my model here, is to acknowledge complicity. I've benefited from microbiome research, from the extraction of fecal samples from the Indigenous community with whom I work. Early on in my career, I didn't frame it this way. I used words like traditional. I talked about their ecology as being similar to our ancestors. I did not pay close enough attention to the ways in which I spoke about the people I worked with. Now I'm going through a phase of reckoning in my career, largely driven by critiques from the Hadza themselves, from my longtime friends and interlocutors. I've been forced to look at ways in which my own practices have been extractive and non-beneficial to the Hadza, and I'm actively working to change that. I've made many mistakes. I'm sure I'll make many more. My entire research paradigm is now working on trying to rectify some of my own past wrongs. We now include community inclusion and or community-based participatory research in every project that we do. I make the distinction because increasingly they're talked about as the same thing. Having a project that includes the community from the outset, from project design on up, is very different than CBPR, community-based participatory research, which includes research that the community has asked for. It's also really important, hopefully people find it important, to engage in more ethical practices by encouraging ethics training for all parties involved. This would mean from the bench to the field to the biobank. And finally, and arguably the most challenging one to achieve, which would really warrant its own hour-long talk, is essentially to engage in benefits and profit sharing arrangements with the study community ahead of time. And this is particularly the case when products are being derived. This is the only way that the threat of biopiracy can be mitigated. So we're far from having a model of how this works, particularly given variation in how Indigenous communities interact with the nation state in their own countries, in their own economies. We can look to Indigenous scholars themselves, however, for how to enact some of these possibilities and some of these agreements. And I specifically point to the work in genetics and genomics highlighted here, including the work of Katrina Claw, Kimberly Sosi, and Keolu Fox, scholars who I have little alert pings on my Google Scholar to read their work when it comes up. I'd like to end my talk today with a quote from a collaborative piece that I'm currently writing with Stephanie Schnorr, Justin Lund, and Shani Msafiri Mongola. And it's a perspective piece that's currently in revision. And we argue in our piece, given how siloed academic disciplines are, it's only through transdisciplinary dialogue, which can often be uncomfortable and force self-reflection, and working alongside Indigenous scientists and participants, can we truly work towards a stronger academy. Rather than being held separate and treated as removed from the process of scientific inquiry, we argue that more ethical research engagement from start to finish should not be held separate from science, but rather is integral to maintaining its rigor. I would like to thank all of my current collaborators, but specifically the ones pictured here, for their science, their collegiality, and their friendship. This talk was inspired and based on our conversations as well as our shared work. And I would also be remiss if I did not thank the foundations that have funded my research. And of course, above all else, an enormous amount of gratitude to the Hadza. They've been putting up with me for a very long time, and I remain very grateful that they do so. And I now am open to questions, comments, all of the above. Thank you very much for listening.
Thank you, Alyssa. Thanks so much for a great talk, really thought-provoking, really important stuff. We have a half an hour for discussion, so I will curate the discussion. If people would like to get in the queue, the best thing to do is use the raise hand function, and I'll keep a list and call on people in order. First, let's take a question from Molly.
Well, I'm first. Well, thank you for such an interesting talk and sharing this perspective, and your work in the area is all great. And I really, I guess, agreed with and resonated with what you were saying about the colonialist kind of rhetoric and how problematic it is. But I was also thinking how, I mean, I have a million questions that it raised, but just to, okay, just like first two things. So one is like, the first one's a comment then, before I ask the question, which is just to add to the list of problems about this rewilding movement, which I wasn't very familiar with myself. Before you talked about it, I was just thinking about how the sort of loss of old friends, you know, however you define it, is problematic because it's thought to be problematic biologically because it's necessary to educate the immune system. And that mostly happens during very early stages of life. And so the idea that you would then intervene during adult life and solve all of your medical problems is not really entirely realistic or quite understanding or appreciating the system that you're intervening in. So that's just one comment. I'm sure that you had a million things that, you know, don't fit into the talk, of course, that I was just going to add to your list. But then I was thinking about how you were saying this idea of like, what is the ancestral microbiome and how we can't know it, but we're not just Homo sapiens, right? We're also primates and we're mammals, and not all of the microbiome research has been in our species. It's also been in these other species. And there is a fair amount that we're learning about the microbiomes of wild primate populations and wild non-human primate populations and non-human mammal populations as well. And there are, like, I wouldn't throw the baby out with the bathwater. Like, yes, there have been problems in the human microbiome research agenda, and I see what you're saying about that. But we also have learned quite a lot about general mammalian and even primate microbiomes that do give us an indication of what an ancestral microbiome might look like, at least in an interspecies sense. And so I wouldn't quite say we don't know anything about what an ancestral microbiome is. And then not only that, but there have been clinical research studies that take that information. For instance, the ubiquity of helminths, which you brought up as like a classic example of something that a lot of people believe would have been, you know, quite a typical part of both the human and, you know, other mammalian microbiomes for a very long time. I guess you were mentioning like the idea of health writ large throughout the talk, but it's a very specific set of diseases that we're interested in addressing through this kind of work. And so, for instance, there have been some really promising clinical trials in multiple sclerosis with helminth therapy. And so I guess, like, I feel like we know a little bit more than maybe you were indicating. Of course, there's like an enormous, you know, world of research that still needs to be done. But what I think is kind of remarkable about this area is that if we didn't have this historical and evolutionary perspective and cross-species perspective even, I don't know what doctor would have come up with the idea to start using helminth therapy to treat multiple sclerosis. You know, it's like, it's one of the wonderful things that I think has come out of this work. So anyway, I guess I was just curious about your comments on all that.
Thank you. Okay, first of all, for the first part of your comment, I wholeheartedly agree. I tried to sneak some in there with my comments on vertical transmission because the other arm of my research is reproduction. And so the other arm of everything that I do involves moms and their babies. And I'm a very big fan of your work. And one of my graduate students, Kristen Herlosky, and I used your work and your helpful comments on some of her presentations at AAPA to inform her work on essentially postpartum maternal mood among the Hadza. So I thank you for that. And yes, I completely agree. Shifting to your question, I must have given off the wrong, perhaps I should have been a little more celebratory of the helminth data, because I completely agree with you and everything that you just said in terms of helminth therapies. And we have some really robust and promising data coming out of clinical trials. So helminth load in particular, I think that this is actually a place in which we see these old friends, and we have demonstrable evidence that suggests that this could potentially be a very impactful and positive intervention. So I'm not including helminth in this argument at all whatsoever. In terms of the species, I'm going to dump a link into the chat thread. This is the most recent kind of discussion that I've been in. We recently had a paper come out in Frontiers in Ecology and Evolution, the internal, external, and extended microbiomes of hominins. And there's a really large discussion in there that specifically talks about the whole body of literature that you were just referencing in terms of what can we actually glean from putting humans in this comparative context. And so my background is in nutrition science, and my background is in diet composition. And so I started out looking, which this might not be relevant, but it might help with how I've come so far from where I started. I started out as a comparative functional morphologist looking at feeding anatomy and adaptations in non-human primates. And I was a research assistant in Adrian Zillman's lab at UC Santa Cruz. It feels like 100 years ago. And one of the things that we specifically talked about is where can you pull out of this, essentially this gradient of diet and adaptations to diet broadly, or however you decide to want to, whatever you want to zero in on. What can we actually pull out of that and say something meaningful about, right? So yes, I think we have learned a lot by looking at other species. And I agree with you. I think that it is short-sighted to only look at humans as primates. As a person who studies diet and studies dietary adaptations to arid savanna environments, I argue that we are not looking at baboons more closely enough. Different talk for a different day in terms of diet composition and social structure. But I agree with you in that regard. I would counter by saying that we know a few things, and we know those few things really well, and we keep hitting those same points over and over and over again. We know that complex carbohydrates, we know that fiber does a number on your gut, period. And we know this, and this is reinforced by all of the beautiful data that are coming out of cellulose-rich diets of non-human primates, particularly great apes. So we know that, and we see it in foragers, we see it in great apes, we see it in lifelong vegetarians, we see all of these things. So if we want to characterize the ancestral microbiome in an ecology that consumes a lot of fibrous plants, then I would agree with you. But just like there is no one Paleolithic diet, there cannot be one ancestral microbiome. We know there's increasingly really fascinating data coming out of communities who consume, so essentially from circumpolar regions, who consume a lot of animal protein. Probably one of the most robust studies is the one that I pointed to in the talk, which is about Mongolians. Their microbiome looks very, very, very different. And even when they eat their traditional diet that is largely based on animal protein, that ancestral proxy microbiome looks very different than an ancestral proxy microbiome of a plant eater. So we also need to pay attention, and for me, in terms of where are we sliding our time scale in terms of the evolution of the human microbiome. The Paleofeces paper that came out just, I think, God, not even 10 days ago, talked about Paleofeces, and they were the samples were 1,000 years old. So part of this is that anthropologists have an opportunity to enter the conversation with a much, I don't want to pump us up too much, but we can enter the dialogue with kind of an understanding of the greater timescale and how humans as a holobiont and how kind of selection pressure can act on humans as an ecological unit. We have kind of an understanding of this timescale in a way that I think a lot of microbiome scientists or microbiome enthusiasts do not. And so they're essentially co-opting a lot of our work incorrectly. So I definitely don't want to throw the baby out with the bathwater. I am certainly not arguing that we shouldn't continue to do cross-cultural and cross-species studies. I think we just need to pay better attention in terms of how we're framing our arguments and which data points we want to pull out to amplify to make an argument. But yeah, I 100% agree with you, and I'm go helmets. Yeah, thanks, Molly.
Thanks.
Nice. Okay, let's take a question from Brooke.
Thanks, Alyssa. It was a great talk. My question is more about the ethics and less about the nitty-gritty of the science. But I really appreciated the whole talk, but particularly, when talking about your own process and reflecting back on your own history as a scientist and what that means in terms of what you've learned and how you've grown and changed how you think about these things. That really resonated with me. And I'm asking a question, I guess, sort of on thinking about the graduate students or people who are about to go into the field for the first time, because one of the things that I've struggled with in terms of how to engage with communities and how to be a representative in some sense, because you are publishing things or you're writing blog posts, or is how to do that responsibly when you're new to a place, right? So there's this sort of competing pressures of coming in, getting a dissertation, getting your name out there in the world and everything, right? And publishing. And then there's also the sort of sense of needing to really be humble about knowing what you don't know when you go to someplace new. And having to take time to build trust with the community and know what they actually think about some of these things, right? Because that doesn't always come, or almost never comes, in sort of primary interactions with people. So I'm just wondering if you could go, now that you've sort of had all these reflections, if you could think back in time to, 15 years ago or something, what your advice might be to people who are just starting to build these relationships.
Whew, that was a doozy, Brooke. Okay, let me see if I can. Okay, I'm gonna tackle it in 3 parts, because I actually think that you are highlighting something that is really important, and you're shining a flashlight into an area, a little dark corner that needs to be talked about. Let's just name it. I'm at a place in my career where I can do this. Let's just tell the truth in this academic circle. I have tenure, I'm going up for full, which maybe in retrospect is not the best time to be, throwing these things out. But here you are. For me, it was, so I'm benefiting from that. That certainly did not drive this shift. The shift was driven, it started, let's see, the first field season where I had some really intense interactions with Hod's of Friends, where we actually dialogued about all of this for the very first time. The very first field season where that happened was 2014. And I most certainly was not in the position I'm in now. So my thinking, my own kind of metamorphosis started long before I guess I had the comfort of my professional career stage in order to do this. So I want to just acknowledge that what you said is very valid, and there's really only one way, and that's for us in these spaces for senior titans in the field, which I see in front of me in my Hollywood Squares here, and for graduate students who are just starting out. In these spaces, it's important to talk about this stuff and to name it, because otherwise, you know, we're not going to shift the discipline. We're not going to shift the academy in general. So I think increasingly we're seeing more open dialogue about how important it is to include the community with whom you work. That is not always possible, particularly when you're a grad student. So, okay, so now I'll shift into the second part of this, which is, I guess I'll answer that by saying kind of what I think about now. I can't go back to 15 years ago. That'll be the third part of my answer, and I'll do it quickly. But this part, I think that there are still ways that you can potentially mitigate human rights infractions, even if you are a short-term visitor. And you and I have talked about some of this with our piece, which was plugged there early on, navigating navigating the waters of cross-cultural research. Even if you are a short-term researcher, there are ways to do this. One of them, if you are NSF, for instance, so will NIH, they will fund trips to return your results. They will fund that. They will actually give you money in your budget to go return your results to the community. You simply have to put it in your initial budget, which often doesn't happen. We don't talk about capacity building, but increasingly, depending on which NSF directorate you're applying to, they really are looking at broad impacts in terms of what this means to focus on things like data sovereignty, to really include collaborators from the study community, collaborators from low and middle income countries who are facilitating your permits, who are facilitating your entry into the country, who are facilitating your access to this community. There is a push to include those individuals from the beginning. So it doesn't just like you land on the ground and all of a sudden like you're best friends with everyone and you start community-based participatory research. It's slow science, which is a problem given the way that the discipline is built, right? It's baked in. You can't really have slow science. So, okay, what do we do? Step by step. My answer is perhaps not the best one, but you do it step by step and you work with every engagement, with every project, with every grant, with every public speaking engagement, as you mentioned, you work, right? And so the first step for me was listening. That's still the most important step for me, is listening to the community I work with. But that took years. I've been there since 2004. It took years for them to actually start talking to me about some of this stuff, which is why I won't do extractive microbiome research anymore. It certainly hasn't stopped me from writing about it, right? I'm still full of all sorts of opinions, but I'm not going to collect those samples anymore. So my third part would be going back 15 years. I was trained in a very different discipline. I was trained in nomothetic data collection, you know, and whether that's good or bad, right? Post-positivist, right? I mean, models here. My training was not this. I myself am an autodidact when it comes to this, because I'm now working alongside Indigenous scholars, and I'm now working on projects that are a bit, they're less in my comfort zone of my original training. And that's scary. But I think we also have to give ourselves the freedom. We have to be able to change as a discipline, as individual scientists. We have to be able to change, and we have to be able to stand up on the mountaintop and say, I made a mistake. I talked about this in a way that was not okay, and I'm owning that mistake. And that's sort of, I guess, where I am right now in response to your question. Is now me wants to tell 15 years ago me to sit down and be quiet and to listen to other people in the room. Now me wants to tell 15 years ago me to start reading scholarship that is not new, that's been around since the 1960s, that has, you know, largely been overlooked in evolutionary anthropology and biological anthropology, evolutionary social science in general, up until very recently. So now me wants to tell old me to keep reading more. Yeah,
Thanks. It was great.
not the best answer, but
I didn't mean to throw a bomb, but that was a great answer.
It's okay. Bomb throwing is okay. It keeps me on my toes, Brooke. And from you, it's all is welcome.
I agree. That was what you just said, Alyssa. I want to second that. Double, triple it. appreciate your pitch for slow science. I'm also an advocate of that. And I mean, I think it's what you've said also, and Brooke's question was so fantastic. You know, something I see for students in bioanthro, but also in all the subfields in our department, when the students are coming in, you know, it's confusing because, you know, how you do research the right way, our standards are changing very, very quickly. And you can't, I mean, I agree with you. It seems like you can't really look to what people were doing 10 years ago as a model anymore. And if I were a student, you know, I would find that confusing because we're asking you to do research and to innovate at the same time, and there's risks involved, right? Because you may be doing research in a way that doesn't look like previous research, and you don't know what's going to happen when you try to publish that. So I really appreciate your pointing that out, and that some of us, you know, who have tenured positions can afford to take those kinds of risks. I remember when I was a postdoc, my advisor, Gerd Gigerenzer, who was a big against null hypothesis significance testing, was one of the things that he would write about a lot. And he said to us, when I was a postdoc at MPI, I dare you to publish papers with no p-values in them. You know, I challenge you to do that. And it was funny because, right, it's like, yeah, I'm not sure I'm going to be able to do that and get a job, or even get my stuff published in a journal. But so anyway, I appreciate you're pointing this out, and as faculty, I agree we should try to, you know, we need to think in new ways about how to support students in their research. So sorry for rambling, but I just wanted to add an appreciative comment. Can we hear from Dan? Dan was next in the queue, and then Maddie, and then Molly.
Thanks, Alyssa. Really, I would say, important talk. And I agree with you on many, many counts. One sort of an empirical comment building on some of the things that Molly said also, which you sort of alluded to. I think you'll agree with this, but I think it, for the larger audience, it bears discussion. And that's, you know, just paralleling the silliness of the paleo diet movement, right? Is the, you know, the notion that, you know, that 20,000 years of coevolution in, you know, the domestication of plants, animals, the you know, bacteria in our gut and our own genomes, that somehow that isn't relevant here, right? And even if it were true that, you know, fecal samples from the Hadza are somehow, you know, directly reflective of what was once the species typical microbiome, that wouldn't necessarily make for a smart prescription for people who are descended from, you know, agriculturalists, right? There's no reason to think that, well, you know, gosh, your skin tone seems to have evolved at a different latitude than the folks whose fecal samples you're collecting, but everything else about you is exactly the same, right? So, I mean, it's just a non-starter from an empirical perspective. And I appreciate your shedding the light, not only on the kind of at the very best, the shallowness of that perspective, but also the frank profiteering of people trying to push product in this. So I imagine you're going to agree with me. I see you're shaking and nodding your head, and you know, I just thought that merited bringing to the fore. But now, consistent with what you telegraphed you expect of me in our earlier chat, let me see if I can push you a little bit. So, you know, we heard recently from Keelu Fox, who you cited as an exemplar, and he perhaps offhandedly suggested that, you know, big pharma making diabetes drugs should be profit sharing at the 50% level with the Pacific Islanders whose information served to inform the creation of those medications, right? It seems to me that it is, and I'm intentionally being the devil's advocate here, right? I'm very sympathetic to the power dynamics, to the exploitation of people in the global South. I mean, I'm very sympathetic to that point of view and that critique, and I think it's important, and I'm glad that it's being discussed. But I want to push back a little bit because it strikes me as arbitrary to draw a distinction between you know, the genomic information in someone's feces or their milk sample, and their answers to a interview, a questionnaire, you know, or for that matter, their participation in any way in research, right? So by the same token, you could argue that, you know, the 40,000 people who participated in the Pfizer vaccine development, you know, deserve a share of the profits, right, from the Pfizer vaccine. I think this is kind of a non-starter, actually, when taken to its logical conclusion, that somehow there's something special about genomic information or biological samples, and that is inherently different from any other kind of participation in research. And moreover, I would say that at least the UCLA IRB would never allow research in which you dangled in front of a research participant the possibility of untold riches. They would see that as coercive, right? They even scrutinize if you have a $500 lottery for participants, right? That is, the expectation is that participants make a decision about their participation based on the costs and benefits to them at the time, and that their contribution to knowledge leaves that context thereafter, right? So I appreciate all the things that the you know, tens of thousands of people who have participated in my research for decades have contributed to my own career success and to whatever little contribution to knowledge I've been able to make. But I don't think that they own that after their participation ends. And if they did, I don't think that any science would really be possible.
Okay, you did your job, Dan. So first of all, thank you. All right, so I want to start at the beginning and address, so I wholeheartedy agree with everything you said initially about this idea that you can kind of take someone out of their microbial, ecological life history, you know, that really, the entire theater, ecological and microbial and, you know, viral theater in which they have been reared, on which they have essentially evolved, and then take that, whatever it is, species or person or what have you, and then shift it. So 100% agree. I tried to allude to a couple of the studies that have been done with diet, with short-term dietary changes. You get a short change, right? Yes, my microbiome is healthy, whatever that means, for 4 days, right? And so then we know that this is actually, it's long-term exposure. And so this is where the really rich and interesting research on how natural selection may have acted on the hologenome, that's where all of this comes in. And that could have been a whole nother talk. Unfortunately, I had to force myself to kind of winnow that down. But you are absolutely right. There's a whole evolutionary argument to be unpacked just there alone. So we'll leave that aside, and yes, I 100% agree with you. And I'll shift to the second half of my answer is going to be in articulating what I see as a difference between compensation and basic research ethics and benefit sharing, because I don't think those are the same thing. And I think that often they get conflated in the literature. And I point to the work of Kimberly Sosi on this exact issue. And I don't know, I don't know what studies Keolu Fox was talking about, so I can't speak to that in particular, but I'm going to put that one on hold and circle back to that comment at the end in terms of data sovereignty and compensation in that regard. So I think that when it comes to the Pfizer vaccine, in many of these instances, we're talking about citizen science. So this is a big hot phrase. You can Google it, you know, for the students who are tuning in, who are interested in this movement. And a lot of the gut microbiome data has actually been driven by the citizen science movement. So this idea that individuals are willingly contributing to the promotion of, to scientific advancements, which is the case with the Pfizer vaccine. I'm sure that participants in the clinical trial would have loved, some type of profit sharing, some stock in Pfizer. That certainly is not out of the ordinary. Certain things have happened. I'm not suggesting that that's the case here. What I'm arguing is that participating in a Pfizer trial is very, very different from donating a breast milk sample with very little understanding of what that breast milk sample will become. And those donated breast milk samples that then turned into a $60 million profit for Nestle and Biogaia is a very different thing than what's happening with Pfizer. And why I argue this is very different is back to the axes of power. So we'll talk about types of data in a moment, but these axes of power. So we're talking about a deliverable. And I think that this is most clearly the case when it comes to product development. So in terms of interviews, in terms of, yes, arguably, particularly what a lot of bioethicists argue and what a lot of Indigenous scholars themselves argue is that even stories, even answers to questionnaires, even interview responses need to be part of this data sovereignty movement so that communities themselves can determine where and how things are shared or stored. This is really tricky with the new push for open science, which I'm a big advocate for. Those types of data require a very special type of treatment, right? But that's where I think just an acknowledgement that we are benefiting in our own careers and our own personal lives from for the participation of all of these people in answering our questionnaires, in, God, all the hods and moms that have let me follow them around and count how many tubers they put into their, conga. I think we start with an acknowledgement and we move forward from there. I'm specifically interrogating product development as it relates to extracted biological samples, which I think is I think we're talking about a wholesale shift. And the reason I think this is because $60 million is not a lot, right? So if you tried to tell UCLA anthropologists can enter into the conversation. We can certainly shift the way that hopefully the discourse goes, but we are not doing the types of IRBs that research in translational medicine is doing. It is a completely different IRB process. And I would also argue that IRB is the lowest of the low-hanging fruit. Making sure that you've met your university requirements for IRB approval by no means means that you have met the ethical standards of the communities that are being sampled. So I think we also have to think about that. So how do you differentiate between coercion and fair compensation? That's a tricky one, and it's going to require a scalpel, and there is no one size fits all approach. And so I think part of this is where there needs to be some type of gold standard best practices that we can look to. And so certainly in genetics and genomics, we are starting to develop those best practices with things like the Nagoya Protocol. There are now, it's built in, right? So now if you are going to be producing some type of product, you must enter into a benefit sharing agreement with the donor community. And here's the caveat, and this is why I'm circling back around to Keolu's comment, which I think this will make sense if I can make the jump here. If those countries participate in those agreements, right? So not every country from which we extract genetic, genomic, or microbiome data is part of the Nagoya Protocol. So this is where it becomes particularly tricky, and where I have learned so much from my friend and my collaborator, Shani Mongola, who has pointed out to me that one of the biggest problems is that we treat all Indigenous communities and their relationship with the nation state the same. And so researchers go into any given research environment thinking that it's going to look one way or the other without any really understanding of how that community has interfaced with that government time and time and time again. So what it means, like for Tanzania, there are no restrictions on this. So even if so it's not illegal. It is not illegal to go into Tanzania. Now they're shifting, practices are shifting. The Commission for Science and Technology is shifting their guidelines, particularly as it pertains to biologically extracted materials. But it's country by country. And so there is no easy answer. And so you essentially have your finger on something that we have to figure out. We have to figure this out. We also have to pay attention to who's being exploited. So marginalized, or people always talk about vulnerable communities, right? But a lot of these vulnerable communities have been made vulnerable. They have been marginalized by their own governments, by the nation state, by researchers. Whether or not we intend to do so, part of this acts to further marginalize them. So I think it's incumbent on us to make sure that our IRBs know that if I'm developing a product, I'm going to get an attorney, and I have a big fancy university to help me with this benefit sharing, profit sharing agreement. Many communities now have, the HODs don't yet, hopefully, but they don't, have essentially their own team, right? They have their own ethics approval office. I know this is certainly true for a lot of Native Alaskan populations. This is starting to happen. This is already the case for San peoples. We're heading there. It's slow, and we've got to work all these kinks out, because you can't just make the wholesale shift and say, all right, 50% of all products are going to go to the community. I think that it requires more nuance, and I think that you need to have culturally appropriate compensatory measures, and that is incumbent upon us, the scientists, to figure out what that means in that grant context, in that cultural context, in that governmental context. So my answer is it's really messy, and it's going to continue to be messy, but we will not affect any positive change or reshape the way that we do our science unless we start trying.
I think this is a longer conversation. I know that there are other hands that are up there. So I appreciate your response. I agree with some of the things in your response and not with others.
Okay.
Okay, I see a comment from Molly. Let me quickly read it.
Molly put some of the text that she uses in her IRB, which specifically informs participants in studies that they will not end up getting any profit.
Right. Thank you for that, Molly. I actually very, very much appreciate you putting that in there, because this is illustrative of part of the larger conversation in terms of how this works. There's also IRB in general is a really sticky issue, and I don't claim to be any expert by any means. I'm part of the NSF methods camp that is kicking off, and I have been tasked with writing the IRB lecture, which is giving me hives just thinking about how I'm supposed to tackle this for students entering into the discipline. So yeah, it's a thorny one. And what's interesting, which I think for me is part of why I appreciate the provocation, Dan, is because we don't talk about this. This is actually something that we really don't talk about in a lot of spaces like this. We just tell our students, get your IRB done. Do whatever you need to do to get that little box checked so that you can get your grant done. And so I certainly never had a dialogue like this. I certainly was not educated in all of this. And so I think that I think what the IRB process looks like for working with Indigenous participant communities, what that looks like 10 years from now is going to be radically different from what it looks like right now. And I think rightly so.
Just a point of clarification, I wasn't referencing the UCLA IRB to hold up the IRB process as an exemplar of the ethical standard that students or anyone else should rise to. Rather, my point was that dangling potentially large compensation, even unimaginably large compensation, is in itself considered coercive, right? And that's precisely why in the medical context initially, high compensation was disallowed. Right. So I think that needs to be part of the conversation. But as I said, there are other voices, so I'll shut up now.
Okay, but I just have to say one thing because I think you're absolutely correct, Dan. I agree with you. And I think that this is why we need to start normalizing iterative and dynamic IRBs, because to say all of that at the outset is preposterous. This is why continued engagement with the community long after your final sample has been collected is necessary. And until we have research institutions and universities that are amenable to iterative IRBs, the process won't get, this won't be fixed, because you're right. You can't just start out. It's not realistic And it most certainly would be seen as coercion. And then you have a whole other, woof, then you're into a whole other ethical, grab bag. So yeah, I agree with you, but I think this is part of why we need to start talking about what iterative IRBs look like.
Great, thank you, Alyssa, and thank you, Dan. Alyssa, can we ask your indulgence to stay for a few more minutes? Okay, because we have a couple more questions. A question from Maddie. Hi, Maddie.
Hey, hi. I'll try to keep it brief. So first off, thank you for doing this. It's been very disturbing to see biological anthropologists acknowledge these issues and then not take responsibility for developing them. So I want to thank you for that. The next thing I want to point out when it comes to the Pfizer analogy is kind of echoing what Alyssa said, that better framework would be something like if high-income nations like ours didn't undermine COVAX or groups like Gavi with bilateral agreements. So that's what I think like a redistributive justice framework would look like for something like vaccines. The second thing, kind of keeping with medicine, you know, full disclosure, I haven't read Yang 2016, Helmrich 2015, or Reed et al. 2019, which were your citations that had to vaguely reference the mania concept, which is what I'm interested in. And I'm wondering if there's really additional conceptual legwork that's done by this concept. I don't know, like, but perhaps you've written about this yourself, and apologies if you have, I haven't read your work. But yeah, because at first I was thinking like, oh, this is like another example of like a gross vestige. Then I started thinking about, you know, if someone wanted to recruit like a biopower sort of framework for talking about this, there are actually some possibilities for making it a more conceptually rich discussion. Yeah, so I have stuff to back that up, but in the to be concise, that's it. And thank you so much for doing this. Yeah.
So first of all, you are not expected to immediately read all of the papers that I use to support my argument. So you are forgiven for not reading 4 papers immediately as I presented them. And I would also, I think you're correct. I think that you are spot on. I think that there is room for a much more rigorous treatment of all of this. I have not come across anyone who's written about microbiomania and kind of the reimagined noble savage paradigm in the context of, in the framework of political ecology of the body or using Foucault's biopower. This was my own brainchild. So I'm thrilled to think, I'm thrilled to hear that you think that that connects together, because that means that the piece I'm writing actually does make sense. So the phrase microbiomania is not very popular, and it is borrowed from applied human geography, specifically in regard, we're talking a lot about helminths today, so specifically in regard to reworming. So in regard to essentially reworming with helminths, which is its own talk in and of itself. But there's actually more science behind, as we discussed earlier, reworming than there is with rewilding, right, with certain bacterial species. So yes, I think that there's a lot of space for this. And I think it's also time for biological anthropologists and behavioral ecologists like myself to start reading more widely and to start reading what human geographers and political ecologists and human ecologists and conservation biologists are talking about. And this is where humans as a holobiont, kind of the whole hologenome concept comes from, is we're not communicating, right? There's not a lot of crosstalk here. And so the ways in which these ideas can be harmful to donor communities aren't really talked about, but I would argue equally as important, because there is not a lot of crosstalk. The science is not waterproof. And that needs to be our starting point for engaging other disciplines. For essentially pointing out these holes, you need to demonstrate that the science is on shaky ground. And then I think we move from there. But yes, I agree with you, and I thank you for the comment.
Yeah, just the last thing I wanted to say, if you're not aware of, or anyone actually, antibiotic-induced mania, that might be something that would interest people, and just two sources that go into that. Thank you so much. Have a good one.
Thank you, Maddie. Okay, we're well over time, but let me, if you don't mind, Alyssa, let's take our final question from Brooke, and this will be, and then we'll wrap up. Brooke.
I just wanted to come back briefly to what you and Dan were talking about, just in that, and sort of what Molly posted actually from her IRB, right? Because when you were talking about, you know, iterative IRB processes, I think it's also just a callback to what you said earlier about just doing the slow science, right? Because you can have an IRB statement, like, now I did genetic testing, for example, and I don't have a statement like that in my IRB. That was not asked of me, but I did have conversations with the community before we started doing this that were specifically about the potential for profit and things like that, right? And I think there is a way to have these conversations with communities where you are distinguishing between the kind of thing that Dan was talking about, where like I run all these studies and there are, you know, my participants are compensated in some non-coercive way to do a survey or whatever. And then that's, you know, that's their contribution and there's no sort of further profit sharing or anything like that, right? But I think that when I've started to talk to people in the community about this, when we started to do this, they really wanted to know the details of that. Like I, you know, said like, yeah, I get a salary off of this, right? My career is being built basically off of the research that I am doing and you are participating in. And so I wanted to be transparent. Like when they are asking me whether there's profit to be made off of this, right? Like that's part of explaining that, like, yes, like there is personal gain, personal professional gain for me, that I think, at least in the conversations I was having, was seen as very distinct from giving your genetics or your, you know, microbiome samples or whatever to, in our case, 23andMe or something like that, right? And so we had to be really careful, you know, that we weren't going to do that, right? And B, to make it clear that we weren't. And actually, I think this is from a previous conversation with you years ago, to make it clear to them that just because we weren't doing that doesn't mean that other people couldn't come into the community asking for the same kinds of samples, but with a very different profit-driven purpose. And I think it just gets to the point of, you know, the IRB being the lowest bar, right, in terms of making sure that the community understands these things and understands them, I think, in a context that goes sometimes beyond your research and what you're doing, but helping them to understand the sort of process at large, because there are these kind of predatory biopiracy kinds of issues going on.
Yeah, thanks, Brooke. I think you articulated that very clearly, and I 100% agree. I mean, I think it's also, you know, we can get into the weeds here because participant compensation is in and of itself a hotly debated topic, because there are some scientists in our discipline who don't want to compensate at all, even for an interview, even for a survey, because that is seen as coercion. And it is a position that's well supported by many of my colleagues. On the other side, I have colleagues, many of them are Indigenous bioethicists, who argue that every interview should be compensated, right? Because you're dealing with these axes of power that you articulate when you are going to, so you essentially get permission for even secondary and tertiary uses of data. This makes it particularly challenging for me with microbiome data, in terms of how, you know, increasingly there are calls, particularly from high profile journals, to make all of your data open access. So what does this mean? How can you actually ensure anonymity? That's just one of 100 different issues that we encounter, and there is no easy answer. And so unfortunately, as I teach my students when we do our research ethics class, ethics are so messy, right? Because we don't have a straightforward answer. You just use the tools available to you, you listen, and hopefully in our position now, I don't know, my goal, I guess, is for my graduate students to be better scientists than me, way better. I want them to surpass me in every possible way. And that means that now that I'm in these spaces, I can hold the space and then seed the space. And I try to seed the space whenever possible to the Indigenous communities themselves and to those voices that are emergent and that need to be heard. And that certainly includes the scholarship of Indigenous scientists who have very strong ideas about this. We now have scientists coming out of communities that have been sampled and studied. You now have Yanomami human rights activists who have a lot to say about community compensation versus benefit sharing. And so I think there's a lot there if we choose to look that I think can be helpful for us in terms of how we integrate it into our discipline so that it is feasible, so that we avoid what Dan pointed out is a very real consequence if we just, kind of rush into this and try to have the same solution for every community and for every researcher community dyad.
Great, thank you so much. Thanks for that question, Brooke, and thanks Alyssa. So I'm mindful of the time. I want to thank you, Alyssa, for being willing to stay over. I feel like this conversation has been really, really useful, and so I appreciate your generosity there. And I want to thank you also for a fantastic talk. This was really great. Thank you for coming to visit us virtually. Hopefully you can come visit in person sometime soon. With that, I'd also like to thank everyone for having been at Beck this year. I've had a great time running it, and I'm really looking forward to what's in store for us next year, hopefully when we're back in real life. And before we leave, Brian, would you say a few words about the format? We have a mixed format in the fall. There'll be an in-person and online option.
Yeah, and I also have to say thanks, Alyssa, for that great talk and wonderful conversation. I spent a lot of time listening, and I appreciate all the experience and accumulated wisdom that you shared with us here. Yeah, so about Beck next year, I'll be following, Clark's great example of keeping our community together amidst these really hard times. So I think we should also say thanks to Clark for doing a great job this entire year. Seeing his face every week has really been the center of our community. So thank you very much, Clark.
Thank you.
For next year, I'm proposing to do a hybrid format, which will provide flexibility for our speakers and for those in our community who might be able to participate in person or virtually. So I'm leaving at this early stage of planning the speakers next year. I'm leaving this as another way to entice speakers to come and join us, is to give them some flexibility on that. But I also really look forward to seeing some folks in person next year in whatever capacity the university makes available to us. So we'll be monitoring the policies that are announced, but so far at least things seem to be trending in a direction where we should be able to be together for a significant fraction of the talks next year in some kind of accommodating form of gathering. So more to be said later as the speaker series takes shape.
Great, thanks, Brian. Yeah, the hybrid model sounds really nice in that it offers multiple options for speakers and for participants. So thank you for getting that together, and we're really looking forward to next year's series. With that, I will stop the recording.